So there are a few people here in the room and a few people on the web watching you by Zoom. So the seminar is by Ben Simons from University of Cambridge. And the title is Theories of Stem Cell Renewal, which is kind of a follow-up to the lecture I gave 2 courses ago. Ben, so it's your turn, and we're happy.
Thanks, Jean. Actually, Jean, can I ask just one more question before I begin? Yes. Which is an important one. How long would be respectable to talk?
How long would be respectable? Like a standard seminars, I don't know, 45 minutes.
Cool. Okay. Very good. Yeah. Just checking.
Is it what you have in mind?
It is what I had in mind, but I didn't want to abuse the audience. So.
No. That's what's expected.
Okay. Thanks.
Okay. So go ahead.
Okay. I'll start. So thank you very much, Jean-François. So yeah. So what I'm going to talk about theories of stem cell renewal. What I'm going to do actually is to go back a little bit in history. And then I'm going to get up to new results. And we're going to look at the old results, perhaps in the context of the new. So let me just begin by defining what I mean by stem cell renewal. So as you know, to replenish cells that are lost through damage, exhaustion, and aging, many tissues constantly renew through adult life. And this process relies on the activity of minority populations of stem cells, which are defined by their capacity to renew, perfectly renew, and give rise to more differentiating progenies. So one of the major challenges in stem cell biology is to understand in the adult context how stem cells function to perfectly balance cell duplication and differentiation to maintain tissue homeostasis. And importantly, if we understand that, we may understand how those programs become dysregulated in the transition to disease states. So let me begin. This is a slightly mischievous slide because I'm not sure anyone really believes in this anymore. But if you go back 20 years or so, if you ask people this question of how stem cells function to maintain tissue, the answer would have been the following, that stem cell function was thought to be invested in functionally discrete cell populations that are individually long-lived. It means that each stem cell by itself has a capacity for long-term renewal, and it achieves that through perfect asymmetric division. And it's characterized by a unique gene expression signature, and those cells sit at the apex of a one-way differentiation hierarchy. But are any of those principles really true? So by the end of this talk, I hope to persuade you that all of those principles are actually unlikely to be realized in a real context. So how can we address the principles and mechanisms of stem cell fate when apparently everything matters? So if you look at what's influencing stem cells, they're responding to secreted factors, signaling factors, different cellular components, physical forces coming from ECM and also from the tissue. So what we have is, in principle, a fiendishly complex problem. So how can we address mechanisms of stem cell fate? Well, as some of you may know, my background is in condensed matter physics. And so, taking a lesson from physics, of course, what we're going to try to do is, rather than trying to map all of this complexity and understand what's happening in a bottom-up way, which may be hopeless, we actually can proceed more effectively by questioning whether stem cell populations conform to some simple types of emergent behaviors, and then to try to use these rules to see whether we can gain predictive understanding into what might be the underlying mechanisms of molecular regulation. So that's the plan. The spirit is very much, if you think about the work that's done by physics-minded researchers on flocking birds, they're questioning what might control density waves in flocks without having to question what might be the interactions that enable birds to signal to each other, or even the genome of the cells that make up the birds. So the outline of the talk, it's really divided, it looks like three, it's really divided into 2 parts. The first part I'm going to review the evidence that stem cell populations are maintained through stochastic cell fate decision-making programs. In the second part of the talk, I'm going to look more closely at the problem of the maintenance of the germline spermatogenesis and see whether we can understand what are the mechanisms that support these stochastic renewal programs. And then in just a couple of slides right at the very end, I'm going to talk about the phenomenon of cell state plasticity, which really calls into question the meaning of stem cell identity. So the starting point goes back quite a long way, and it relates to the maintenance of the mouse skin epidermis. So the mouse skin epidermis is a compartmentalized tissue that's comprised of hair follicles, sebaceous glands, all interspersed with interfollicular epidermis. And the interfollicular epidermis is a squamous stratified epithelium. All cell division takes place in the basal layer. And as cells commit to terminal differentiation, they delaminate from the basal layer and they move up through the suprabasal layers, maturing and forming the essential barrier function of the skin. So what is the identity of the renewing stem cell population, and how do these cells function to maintain tissue? So this turns out to be a surprisingly controversial question. So some years ago, quite a few years ago now, we worked with Phil Jones's lab in Cambridge and a very bright young theory graduate student by the name of Alon Klein to study this problem using clonal lineage tracing. And in this approach, what we can do is in a dose-dependent manner, we can activate a yellow fluorescent protein in cells of the interfollicular epidermis. And that provides an indelible hereditary label that will mark that cell and all of its progenies. And by titrating down the dose so that we have a small number of these cells and their progenies are called clones, then we can generate events like this. So this is a clone that has one basal cell, another suprabasal cell, and this is the sort of flattening squame cell which is close to the skin surface. And so what Phil's lab did was to build up the ensemble of clone sizes. And what did they find? So what they found was that the clones from this tissue showed immensely variable fates. So what you can see here, this is a section through the basal layer of the skin epidermis. And here at 1 week, you can see a clone with a single cell. Here at 2 weeks, here's a clone with 2 basal cells, and so on, until eventually at 1 year, you can find clones that have a very large footprint on the basal layer. And here in the inset is that distribution of clones at 26 weeks after induction. And we've got some clones with just a couple of cells and other clones that have between 33 and 64 cells. So a huge variability. But what we also see is that the average basal clone size is continuously increasing, and it increases in a linear-like manner. And that continual increase is compensated by loss of clones. So the number of surviving clones is gradually diminishing. So the overall size of the labeled population remains constant. So where does this variability come from? Is it that we've got some progenitors that have an ingrained difference in their renewal potential, and they know that from the outset? Does it come from spatial heterogeneities in the dermis, which might favor cells in certain regions? Or does it come from the chance fate outcomes of an otherwise equipotent renewing cell population? So what we noticed is that if we look at this distribution of clone sizes, which is very broad, here's the number of cells per clone, each dot is a single clone, then this data over time showed convergence to a statistical scaling behavior, in which although there was an average increase in the size of clones, the chance of finding a clone of some multiple of the average, well, that remained constant over time. And the scaling function in this place maps quite well to an exponential line distribution. So this suggested that although we have this huge variability, the renewing population was undergoing stochastic fate behavior, within an equipotent population, obeying a common kind of statistical rule. So what's the origin of scaling? Well, when we began this project, most people in the field were minded to think that progenitor cells or stem cells in the skin make a sequence of asymmetrical divisions, divisions resulting in asymmetric fate outcome. But the clonal data told us that that couldn't be the case. There had to be cells that could duplicate and cells that differentiate, but those probabilities have to be balanced so that overall, the total number of progenitor cells or stem cells remains constant over time. And those differentiated cells then delaminate. So what kind of dynamics is that? Well, if we look at any given clone, then over time, it might be lost just by chance production of differentiated cells, or it may expand over time by chance duplication events. And you can think of the dynamics as being a little bit like the dynamics of surnames in a population. Each clonal mark, each hereditary mark is like a surname, and that surname is carried by progenitors. If they differentiate, it's like giving rise to girls, or they duplicate to boys. And so if we look at the dynamics over time, it's the same kind of dynamics that you would see in a population of surnames. Some surnames would emerge as abundant and other surnames would die out. So if we focus on the progenitor cell population alone, then that's just a very simple kind of dynamic. It's just a critical birth-death process in which cells either duplicate or the progenitor is lost through differentiation. And that happens like a coin toss with 1/2 probability. So then we can do just a little bit of maths. And if we take this critical birth-death process, then over time, the average size of the surviving clones grows linearly with time. The probability that clones survive drops like 1 over time, and that makes sense because the product of these two better be unity. Total number of marked cells, progenitor cells should remain constant. And the chance of finding a clone with more than n renewing cells, this cumulative distribution, well, that forms this exponential-like distribution as seen in the data. In other words, we converge to statistical scaling with a scaling function which is exponential. So we went back and fit the clonal data. What we found is actually the frequency of asymmetrical divisions is rather high. So some 4 out of 5 divisions result in asymmetric fate outcome, but 1 out of 5 divisions result in this coordinated kind of loss replacement process. But this raises a puzzle. So what enforces this perfect fate balance in the tissue? How is that regulated? Well, it's been long known that stem cells are massively influenced by their environment. And early on, when people were thinking about asymmetric fate outcomes, emphasis was placed on this kind of niche-based regulation. The idea was that factors released by somatic niche cells would maintain stem cell competence. And if stem cells divided, one of their daughters would end up being positioned away from the niche, and remote from the niche, it would lose stem cell competence and enter into a differentiation program. So we can ask, well, what would happen if the niche wasn't discrete and localized like this, but suppose the niche was extended? So if stem cell competence were linked with an extended niche, then the physical size of the niche would be sufficient to control stem cell density. So the idea here is that the stem cells lie anchored to this niche, and as they divide, it's crowded, and some get displaced from the niche. And so if we interpret that in terms of fate outcomes, we can have all possible events of duplication, asymmetrical fate outcome, or 2 cells leaving a kind of terminal division event. But on average, we would have balance because the size of the niche is fixed. And we can ask, what kind of dynamics is that? Well, this process, which we call neutral stem cell competition for niche access, over time, you can show that this leads to a scaling clone dynamics. And that scaling dynamics is in in physics parlance as in the universality class of the voter model. So if we look at what emerges from that kind of voter model dynamics, then in the case that we're interested in of the skin epidermis, that's the two-dimensional case where we expect the average clone size to grow approximately linearly with time. In fact, there's an additional logarithm here coming from the usual the logarithms you get in 2 dimensions. In a one-dimensional context, we actually have a different kind of scaling function, which is Gaussian-like, and the average clone size increases as the square root of time. And above 2 dimensions, that's above the critical dimension, we end up with a mean field, and that's like this critical birth-death process. So that seemed to explain naturally how you would get that kind of dynamics in the skin, that you belong to this voter model class. And in fact, that kind of idea was promoted very recently in beautiful work by Valentina Greco's lab in collaboration with Alon Klein a couple of years ago, where they proposed that the basal cell layer of the skin epidermis constitutes an extended niche in which chance differentiation from the basal layer and delamination promotes the symmetrical division of neighbors. And they built that argument based on beautiful intravital live imaging work in Valentina Greco's lab. So you can see here, this is time 0, this is 24 hours later on. And by mapping all of the events, they built up this kind of argument to support this process of loss driving replacement. And in fact, this idea echoes an idea that came 50 years ago in groundbreaking work by Charles-Philippe Leblond, where he used a primitive radio labeling type of approach in those days of rat esophagus to propose exactly this kind of dynamic. Now, as beautiful as it is, I'm going to undermine this a little bit later on, but let's stick with this for now, that this would explain very neatly how you get that kind of scaling dynamics. And perhaps in the context of the course that this talk is associated with, I should emphasize here actually that there's a long history of really elegant statistical theory around these kinds of models in this context, which goes back to beautiful work by Boris Schreiman, the host actually, Edouard Hanezo, and many, many others. So let's run with this idea of niche-based regulation and look at another tissue. So here is the intestinal epithelium. So in the small intestine, the epithelium is divided up into large villi that invade into the gut lumen, and these crypt structures that invade into the stroma. And stem cells in this tissue are located at the base of the crypt. And as they differentiate, they give rise to progenitors, secreting and absorptive progenitors that move up the walls of the crypt and then terminally differentiate into the functional absorptive and secretory cell types that make up the intestinal epithelium. And those cells move up through the villi where they're shed. And in mouse, this whole intestinal epithelium is turned over once every 5 days or so. So this is a very rapidly cycling tissue. Now, in this case, competition for niche access leads to a clonal dynamics in which we have labeled cells which produce these ribbons of mark cells that compete neutrally around the crypt base. And as they compete neutrally, this boundary of this ribbon expands and contracts in this one-dimensional loss replacement type of dynamics. And so working with Doug Winton's lab in Cambridge, and again, Alon Klein, when he was a student in my lab, we mapped the clonal dynamics in the small intestine. And sure enough, we found that the clonal data converged to this scaling behavior with a scaling function, which was characteristic of one-dimensional loss replacement or one-dimensional neutral dynamics with a square root increase in the average clone size. So the first message of this talk is that when we look at these epithelia and many cycling epithelia, we find that stochastic stem cell renewal programs emerge as a conserved feature of adult tissue maintenance. So here I've made a list of all of the tissues that I could find to date for which that kind of scaling clone dynamics has been found. And in short, everywhere that people have looked for that, this is what they found, that tissues are maintained by these stochastic stem cell renewal programs. But there's a puzzle. And the puzzle is quite an important puzzle. It's, of course, a very simple idea that competition for niche access is easy enough to maintain tissue homeostasis, a very simple kind of principle. But unfortunately, in most epithelia, and perhaps all, the stem cells are not clustered together in a localized niche, but those cells lie scattered amongst their differentiating progenies. So the situation is actually exemplified by the airway epithelium, where you have small pyramid-like stem cells which lie anchored to a basal layer. But those basal stem cells give rise to differentiating cells, club cells and ciliated cells. Club cells are still have the capacity to divide, and the ciliated cells are largely terminally differentiated. And the stem cells are separated from each other potentially by their differentiating progenies. They're not clustered together in some local niche environment. And yet we still find that stem cells adopt a balanced stochastic fate. So they're able to regulate the balance between duplication and differentiation. And this raises the question, how in such an open niche, sometimes people call this facultative, how in such an open niche, when stem cells are separated from their partners by differentiating cells, how do they control the balance between stochastic cell duplication and differentiation? So this brings me to the second part of the talk, where we're now going to drill down on more recent work, but we're going to drill down a bit more deeply into the problem of spermatogenesis. Okay. So in mouse, sperm production, or in mammals in general, sperm production is maintained by the activity of spermatogenic stem cells. And those stem cells lie anchored on the basement membrane of seminiferous tubules. And those stem cells divide. And as they divide, they go through something like 10 to 12 rounds of mitotic cell division before cells enter into meiosis, and they translocate across tight junctions between Sertoli cells, where they then undergo a process of maturation before they're released into the lumen as mature sperm. So we have this very long differentiation hierarchy. We also have this characteristic behavior in which the cells make incomplete divisions. They produce these syncytia, which are connected by interconnecting bridges. And although we don't have, we'll see why, we don't have very good marker identity, we find the stem cell population is enriched in the population of GRF-alpha-1 expressing cells. As they begin to differentiate, they upregulate neurogenin 3. And then when they transition into becoming KIT positive, those cells are then on what looks like an irreversible differentiation path. But another curious feature of the system, which will also have a role, is exemplified here on the left. So on the left here is a movie, I'm going to play it in a second, which shows GRF-alpha-1 positive cells marked by GFP. In fact, those cells, these little dots here, those cells are actually in a crowded environment surrounded by all of their progenies. But all of these spermatogonia are actively migrating. And this is an intravital live imaging movie taken from our collaborators lab, Shosei Yoshida in Japan. And here you can see something like 3 days of continuous live imaging in vivo, where you can see those cells actively migrating, almost foraging on the basement membrane of the seminiferous tubules. So what is the identity of the stem cell compartment? And how do those cells function to maintain long-term homeostasis? How do they do that? So again, we can make use of lineage tracing. And in fact, we did that years ago, again in collaboration with Shosei Yoshida and Alon Klein. So once again, if we mark cells in that population and look at their clonal dynamics over time, we find that the clone size distribution converges onto this hallmark scaling behavior. The average clone size grows like 1 square root of time, and the number of clones per testis is forever diminishing, so the overall population remains constant. And what this showed us was that once again, we have this neutral loss replacement, but now in the context of a one-dimensional tissue. So this scaling of clone size behavior indicates that whatever these stem cells are, whatever is their identity, the duplication of these cells is correlated locally with differentiation. So we have, again, this kind of loss replacement dynamics. So this is just a perfect example of what I said a few minutes ago. With the stem cells separated by huge numbers of their differentiating progenies, vast numbers of differentiating progenies, how do these stem cells correlate locally cell duplication with differentiation? That was the challenge that we set out to try to understand. Let me just emphasize again. So all of this work is done in very close collaboration with Shosei Yoshida at the National Institute of Basic Biology in Japan. So towards a hypothesis. So the first thing that we knew from lineage tracing studies is that all stem cell function appears to be contained within the GIRF-alpha-1 positive cell population. That doesn't mean that all GIRF-alpha-1 positive cells are stem cells. It means that the stem cell function seems to be largely within that population. If we mark 10% of those cells and then look 6 months later, we find that 10% of those cells are still labeled. The second thing is that GIRF-alpha-1 positive cells show some tendency, some bias localization towards the vasculature. So although they're actively migrating on the seminiferous tubules, they tend to be more likely found in these interstitial regions close to the vascular regions. And the second thing that Shosei's lab noticed is that FGFs, which are secreted by these somatic lymphatic endothelial cells on the outside of the seminiferous tubules, those FGFs diffuse through the basement membrane and are then internalized by the stem cells. So you can see that here, FGF5 is marked in magenta, and GIRF-alpha-1 is robustly expressed on the surface of cells, and you can see the FGFs being internalized through endocytosis into those cells. So why does this matter? And it matters because what Shosei's lab observed, and this is work that was driven by Yu Kitadate in Shosei's lab, was that by manipulating the levels of FGF5, we could adjust the steady state density of the GIRF-alpha-1 positive population. So here is the control. And if we knock out one of the alleles of FGF5, the steady state density drops. If we knock out two, it drops further. And if we add a copy of FGF5, the stem cell density, or the GIRF-alpha-1 positive cell density, it increases. So it looks like the levels of FGF5 modulate the steady state density of the stem cell population. And this led Shosei to frame the following hypothesis. And the hypothesis is the following, that FGFs released by these endothelial cells function as fate determinants, and they enable the stem cells to sense their local density and adjust their fate bias in response. So, so the idea here is the following. So when cells receive FGFs, if they receive an abundance of FGFs, they suppress the expression of differentiation licensing factors. So when cells are exposed to a differentiation signal, in the germline it's retinoic acid signal, those cells are not competent to differentiate. But if the environment becomes too crowded, the level of FGFs is diminished because the FGFs are consumed. differentiation licensing factors can turn on, and then cells become primed, licensed, but not committed to differentiation. It's only if they receive a secondary retinoic acid signal that they're driven into this committed state. On the other hand, if they recover access to FGFs, they can revert back to this state of renewal. So one way to think about this as an analogy is that stem cell differentiation becomes a two-stage process in which the factors that control differentiation licensing, the FGFs, are distinct from the signals that drive commitment. It's a bit like the manual transmission of a car. So the FGFs are controlling the clutch, and retinoic acid is the accelerator. When FGFs are abundant, then the clutch is pressed down, and the accelerator can't act. But if FGF levels are able to, if FGF levels fall, the clutch is released, and then retinoic acid signal can move the car. So what's the evidence for this kind of feedback mechanism? So here we worked with a postdoc of mine, a former postdoc of mine, David York, and David built a minimal model to describe what was going on. So imagine that stem cells can duplicate or they can differentiate, but they do that with a probability that depends on local FGF density. Well, this is mean field, so we're looking at a zero-dimensional situation. And let's make that probability like a kind of Hill function. So when F is larger than F0, it means that the stem cells are biased towards duplication. But when the concentration is less than F0, they're biased towards differentiation. Now let's add to that the dynamics of FGFs. So we'll suppose that FGFs are produced at a constant rate mu, a production rate mu, that they naturally degrade at rate K, and they're consumed at rate C, but that has a plateau which is, or it's moderated by the same kind of Hill function. So what kind of dynamics does that, mean field dynamics does that translate to? And it leads to the following kind of stable dynamics in which the stem cell density above a threshold achieves a steady state, which scales linearly with the FGF synthesis rate. So providing the net production rate is above a threshold, we have a stable steady state in which any deviations from that steady state will drive us back into homeostasis. So that's prediction one. So prediction two, if we look at the phase diagram of this system, so here's the FGF synthesis rate, here's the degradation rate. If the synthesis rate is too low, we can't sustain a steady state stem cell density, but above that threshold, we have homeostasis. And then we can find two phases of behavior in which if we knock the system out of homeostasis, it has a monatomic recovery back to steady state. And another phase where if we knock it out of homeostasis, the recovery is oscillatory. And it's easy to think about that oscillatory recovery. Imagine that we ablate stem cells. Now we have a huge abundance of FGFs. So the stem cell population will expand very rapidly. But then it may overshoot. And now there's a crisis because there's not enough FGF. So now the density collapses, and so the process continues. And we can see that in this animation. So if we simulate that kind of dynamics in the two-dimensional setting, then I don't know whether you can see that, but the white cells here are the stem cells, and the cloud is the FGFs. And you can see that kind of oscillatory dynamics emerging there. In fact, we also found phenomena like, excitable behavior and intermittency in this kind of dynamic. So prediction two is that this provides us with a stable mechanism of stem cell density regulation, which affords a signature phase of damped oscillatory recovery during steady state. And then finally, we can turn to the clone dynamics. So if we label cells in a particular color here, and we allow the system to evolve, then obviously we have a process in which clones may be by chance lost through differentiation and others expand. And it won't surprise you that this model, again, translates to that kind of scaling dynamics that we saw before. And so what you can see here is that at short times where the system looks two-dimensional, we have a linear increase in the average size. But once the clone extends across the circumference of the tubule, further dynamics becomes quasi-one-dimensional, and you cross over to this square root T behavior and this Gaussian-like scaling behavior of clone size. So that's prediction 3. We should converge to a scaling clone size behavior characterized by local stem cell loss and replacement. So what do we find experimentally? So we've already seen from the earlier study, here's a more recent study of the clonal dynamics based on GIRF alpha-1 expression. If we look at how the clones expand over time, we have a heterogeneous size distribution of clones. But when we look at the cumulative fraction, the data collapses very nicely onto this scaling behavior with this Gaussian-like scaling function. And the average clone size grows as the predicted T to the 1/2 power. The second prediction was that the steady state stem cell density should scale linearly with the FGF synthesis rate. And what you, Kitadit, did was to look at numerous transgenic animals with different combinations of FGF concentration. And what you can see is that the steady state GIRF alpha-1 positive cell density does indeed scale surprisingly linearly with a total allele fraction of FGFs, very much in support of this very simple kind of feedback model. But perhaps the most striking observation was regeneration. So when we chemically depleted the stem cell density by administration of busulfan, where we removed 90 to 95% of the stem cells, we then looked at the phase of recovery. And the recovery goes through this phase of damped oscillatory dynamics through the recovery phase. What you can see here are 2 different transgenic animals, one in which FGF5 has been deleted. And in both cases, you get this long oscillatory dynamics with a period which runs to more than 20 days or so. So this kind of oscillatory dynamics is really a signature of that kind of feedback density regulation. Okay, so what follows from this is that the stem cell population is not invested in a discrete, molecularly discrete population, but it's shared across states of distinct transcriptional and morphological states. So stem cell function is invested in populations which are enriched in the expression of PLVAP and other factors. But we also have stem cell capacity in this licensed population, which are enriched in other factors. And this licensed population includes syncytial cells, which are pairs. And those cells are able to move reversibly between states that are biased for renewal and primed for differentiation. And it's only the retinoic acid signal that's able to drive these licensed cells into a phase of commitment. Well, is there further evidence to support this? Well, recently we've looked, or Chaussay's lab has looked at lineage tracing using targeted promoters which are enriched in the renewing population or in the licensed population. And this again provides direct evidence for fake priming and reversibility. And in fact, what we find is that the states over here are enriched for renewal, whereas the states over here are biased towards differentiation, but all of these states can move reversibly. And we also find that these states have a very slow division rate, which is synchronous with the epithelial cycle of the seminiferous tubules, and that the transition from this state back into this state involves a process of syncytial fragmentation. This is very preliminary, but we've already looked at published single-cell RNA-seq data, where again we can see evidence for this kind of fate priming and reversibility. So we can see this is the population which is biased for renewal. This is the population which is licensed or poised for differentiation. But if we look at the trajectory through those populations, we can find evidence for this reverse transition. Another test of this kind of model is that we would predict if we inhibit retinoic acid signaling, we should be able to trap cells in this kind of renewing state. And actually what's shown here is we've taken GFP positive cells from GFP positive stem cells, or GF alpha 1 positive cells from a donor and transplanted them into a sterile host. And we've asked about the transplantation efficiency. And what we've shown here is that if you treat those with a retinoic acid inhibitor, we trap cells in that state, they're unable to differentiate, and this massively increases the transplantation efficiency of those cells. So if we block retinoic acid signaling, we are able to enrich, maintain cells in this renewing state. And a third phenomena that emerges from this is it gives us a way of understanding why de novo gain of function mutations in FGF receptors are able to confer a competitive advantage. So by having more receptors, those cells are able to increase their intake of FGFs even if the concentration levels become lower. And that gives them an advantage which leads to this process known as selfish selection that allows those clones to undergo non-neutral clone expansion. And what you can see here is a section through human testis, which shows individual clones which carry mutations in FGF receptor 2 or FGF receptor 3, which are linked precisely to this de novo gain of function mutation. And this phenomenon, it actually helps to explain the increasing incidence of Apert syndrome, which is a congenital disorder linked to a point mutation in FGF receptors in the offspring of aging fathers. So point mutations lead to a clonal advantage, which with age lead to the colonization of tissue. So let's step back a bit and offer some perspectives on this feedback mechanism of density regulation. So you may already have seen that this mechanism is very much analogous to quorum sensing in bacterial populations and other ecological settings. And it is a kind of example of this famous Malthusian trap, which goes back to Thomas Malthus back in the 19th century, where he argued that the size of a population was entirely naturally limited by allocation of resources. So you can see in this cartoon, if you would ask yourself, how many cows can you support in a field? It's not the number afforded by physical contact, but it's regulated by grass production. So if you have a higher rate of grass production, you can happily support more animals. If you have a lower rate of grass production, you can support less animals. And that's what's happening in the stem cell compartment. The grass here, the grass is the FGFs, and the FGF production rate is managing the stem cell density, and the system is able to naturally respond to that density. This mechanism is very robust. So it ensures that density regulation is robust to potential spatial heterogeneities that might exist in the abundance of those niche-like factors. You could think about that as having regions where the grass production is higher, like here, where you can support more cows. But naturally, the animals will sort out that heterogeneity. And it's also robust to temporal changes, as we've seen. It functions as a kind of attractor state. It also extends the range of states that can be mobilized in response to injury. So all of those states, the large number of states that are in this primed condition on injury, can immediately be remobilized back into the renewing compartment. And actually, this mechanism also mirrors a reciprocal feedback mechanism that was recently proposed by Uri Allon. In fact, it was actually slightly before our work, frankly, but we didn't know about it at the time. A reciprocal feedback mechanism to regulate population balance. So to make an analogy, It's a bit like our mechanism, but instead of the niche cells being inert and just producing the niche factors, you can think of the niche components as being proliferative cells themselves that receive factors that are released by the stem cells. So what Uri Alon had in mind here was the balance between fibroblasts and macrophages, where both populations receive and consume signals that are released by each other, which again gives rise to a very stable, stable population balance. And of course, the other nice thing about this mechanism that I can't go into right now is that if you couple this with the ability of those niche-like cells to move around by coupling cell fate with such cell movements, it provides a basis also for self-organization. So if we look at a problem like branching morphogenesis, it looks like what's happening in the tips of those cells is very much linked to this kind of competition mechanism. Okay, so if we go back to that straw man folklore that I had at the beginning, stem cell function was thought to be invested in discrete stem cell populations, individually long-lived, characterized by unique gene expression signature and the apex of a one-way differentiation hierarchy. Well, at least in the context of the germline, all of those seem not to be true. And rather, in testis, stem cell function is not restricted to a discrete pole. It's not characterized by a unique gene expression signature. And the cells are at the apex, but they're at the apex not really of a one-way differentiation hierarchy, but in which cells are able to move around. They're heterogeneous, and they can transfer reversibly between states that are biased for renewal or primed for differentiation. So that's my message number 2. So how generic is such an open niche organization? So we saw before that it was proposed that the stem cell density in the skin epidermis was controlled through competition for contact with the basement membrane. But is it really so simple? Well, if we look at this, right? So this is clonal dynamics in the mouse esophageal epithelium. So as with skin, the clonal dynamics in esophagus has exactly the same kind of squamous organization. It's also characterized by statistical scaling. Here's the basal clone size. Here's the cumulative size distribution. You can see that over time, it's converging on an exponential distribution. And here's this linear-like increase in the average size. So all of this is consistent with this very simple one progenitor model. This model at the basement membrane is the niche cells delaminate and then are replaced by duplication of neighbors. But any model based on balanced stochastic fate will conform to long-term scaling, any model. Now, if we look carefully at this data, right, so if we look carefully at this data, then what you can see is that at 180 days, half a year post-labeling, here's the clone size distribution. And if you look carefully, you can see that clones that have 2, 4, 6, 8, 10, and so on basal cells are much more enriched than those with an odd number. It's true at 84 days. And there's a durable, even odd correlation in the data that in fact reveals proliferative heterogeneity. And I don't have time to go into detail, but working with Cedric Blancpain's lab, we found that even odd correlations in backskin actually show that the squamous epithelia in general, stem cell differentiation is not direct, but it occurs through the terminal division of a committed progenitor cell state. Right? So we have this kind of balanced stochastic fate, but instead of cells going directly to terminal differentiation, they enter into a state where they go through one round of terminal division before delamination. So why is this important? It means that in the context of squamous tissues, as in testis, this balanced stochastic fate, it's not made in the context of an open niche. Sorry, it's not made in the context of a closed niche in which all cells are stem cells on the basement membrane, cells leave and are replaced. It's made in the context of an open niche where stem cells share that environment with their differentiating progenies. And we think it highly likely that in this context, the competition for fate determinants is playing a role in the regulation of stem cell fate choice. And similarly in the gut, intravital live imaging in work that were beautiful work done by Jacco van Rienen's lab and with Edouard Hanezo, intravital time-lapse imaging shows that there is retrograde flow in the crypt that allows stem cells to adjust reversibly between being positioned in the niche border where they're biased for differentiation and loss, or at the niche based where they're biased for renewal. So that bias is linked to subtle changes in transcription. And again, it leads us to question whether this kind of heterogeneity and reversibility is a ubiquitous feature of epithelial maintenance. So this leads me to the hypothesis that the reversible transition between renewing and licensed states, I think, may be a generic feature of stochastic stem cell regulation in renewing epithelial tissues. So it remains to be seen where all of these examples where we find stochastic fate are actually conforming to that same kind of competition mechanism. So I'm almost done. Is fate commitment itself reversible? So I just have really one slide on this, and I have one slide not because I think that quantitative approaches have something singular to offer, but because I think it's so important. So the last point that I'll make is the following, that, that normally people have thought that the only cells that really have a strong regenerative capacity are stem cells. But in the context of the gut, for example, it's been found that progenitors normally committed to differentiation are readily able to reacquire adult stem cell function in response to injury. And that's true both of progenitors in the secretory and the absorbative lineages. So even when cells have entered into what looks like commitment to a differentiation program, they're able to reacquire stem cell function. And that kind of natural cellular reprogramming seems to be ubiquitous across multiple epithelial tissues, including trachea, in mammary glands, and also in liver, and others perhaps as well. And the second thing is that this process could be direct. It could involve a direct transition from progenitors back to the stem cell compartment. But it also can be mediated by natural cellular reprogramming to fetal-like states that seed the process of regeneration. And in very beautiful work by Kim Jensen and Stefano Piccolo, they've actually provided very good functional evidence that, that injury in the gut can drive reprogramming back through the reacquisition of fetal-like states. So my message number 3 is that in epithelial tissues, commitment to differentiation itself may not be irreversible, but cells on a differentiation path, a path where they normally will differentiate and be lost, can naturally reprogram in response to injury. And the important thing here is that this exposes a vulnerability that can, and it looks like it is, exploited in the transition to disease states, and particularly to cancer. And that's very much an emphasis of the work in our lab at the moment. So I'm finished. I should just have acknowledgements. Well, I mentioned a lot of the people are on the way through, but I really should put a strong emphasis on Shosei Yoshida. So all of the work that we've done on germline has been done in very close collaboration with Shosei Yoshida. And that beautiful feedback mechanism that I proposed, I can say that with some humility because that was really Shosei's idea. Shosei really worked that out. So the experimental work on that was carried out by Yu Kitadate in Shosei's lab. And I wanted also particularly to emphasize Alon Klein. All of those ideas were developed, all those early ideas were developed very much being driven by Alon when he was a student in my lab. And I mentioned David Jorg, who was instrumental in doing the theory work, the more recent theory work on the on spermatogenesis. So let me stop there. Thank you.
Thanks a lot, Ben. Maybe I don't know if I can do that. Yeah, ask people on Zoom whether there's someone having questions. If you raise your hands, you can ask questions to Ben. So none of them is raising their hands now. Are there questions in the room? With Pierre. Yeah, okay. I'll repeat the question if you don't hear.
I had one question about the relevance of the spatial diffusion of FGF.
Yes, yes.
And maybe related to this, I'm not sure I understood why this turns out to be a 1D process.
Oh yeah, okay, thanks. Actually, both excellent questions. You might think that FGFs would readily diffuse, and actually we assumed that too. In fact, the FGFs hardly diffuse at all. So they're able to go through the basement membrane, but they don't diffuse very much at all in the context of the tubule. What's actually essential here is that the stem cells are actively migrating. So that's really crucial. We don't need the FGF diffusion if the stem cells migrate on that background. So that's point number one. The second point is, where does one dimensionality come from? And I was probably a bit hasty with that, apologies. So the seminiferous tubule, if you ask how many stem cells there are around the circumference on average, it's maybe 5, 6 or so stem cells per circumference field, right? So what that means is that initially the competition will be rather two-dimensional, but once a clone has wrapped itself around, colonized around a circumference, the subsequent dynamics of the clone can only occur along the seminiferous tubule. And that leads to a quasi one-dimensional dynamic. So that's why we cross over to that one-dimensional type of behavior.
Hi, could you just comment a little bit on the spatial correlation bit, which you roughly mentioned for the Voltaire model, especially in the context of the epithelial tissue and the intestine, where in intestine there is a discrete set of pits, and in the epithelium there is a continuous layer.
Yeah, so in the interest of time, I went through that a little bit quickly. So I mean, there are a couple of things to say. In terms of the competition model, we've certainly not proven, or I mean, as much as one can prove anything in biology, we've certainly not proven that that's what's happening in the context of the skin epidermis. Let me just say that from the outset. What I believe is that if you have stem cells that maintain homeostasis in an open niche environment, that means they share that environment with their differentiating progenies. I don't believe that there is another mechanism that can work that is not otherwise linked to that kind of feedback mechanism. Now, it could be chemical signaling, it could be mechanical signaling, but I think it has to be that kind of feedback mechanism. Now, I rushed through a little bit the epidermis example. That even odd effect, the way to think about that, and then maybe, okay, so the way to think about that is the following. Imagine that the tissue was built up out of discrete units, which were made up of a stem cell and a progenitor. And that when the progenitor terminally differentiates and is lost, it's replaced by the stem cell in the same unit. That would mean that all of the clones were either of size 0 or 2 in the basal layer, okay? So now imagine that occasionally you can have a loss replacement event in which you lose the progenitors altogether from a unit. Then it will be replaced. And when it's replaced, it means that you will have an enrichment of clones that will have 2 or 4 or 6 or 8 cells. And the fact that we see those robust, even odd correlations in the data tells us that those features must be very strong. If we ask what's the origin of those compartments, I've got some ideas about that, but the truth is that we don't know. It's really intriguing. But what it tells us is that the fate decisions of the stem cells, those fate decisions, the choice between duplication and differentiation, that is a decision which is 100% made in the basal layer. It's not linked to cell delamination, it's made entirely within the basal layer. So it tells us that that must be an open niche-like environment. Then it raises the question of how we get that kind of balance. And what I think is its feedback. And then the last thing is the intestine. So yes, so the intestine, so these retrograde flows are really interesting, right? So you might, we've known for a long time that cells at the border of the niche are able to reacquire position at the base of the niche and restore their fate bias towards renewal. We've known that for quite a long time, but the recent discovery is that that process is not a passive process of cell rearrangement, but it looks like it's an active process of retrograde flow, that the cells are actively moving back into that region. And what it's suggesting, I think, is again, maybe one has that kind of feedback mechanism operating there, but that's pure conjecture.
Do you think that your open niche mechanism is extremely general for stem cells? Should I completely forget about closed niche, or would they exist in other contexts?
Well, I mean, I'm going to stick my neck out here, so it's of course I don't know, but let me answer that in the following way. Yeah. So frankly, I think because of my background in physics, I find it irresistible not to think that there are multiple different ways of doing things, that there's not one mechanism that does it. And honestly, I think this kind of feedback mechanism is probably pretty generic for the renewal of epithelial tissues in general. It gives something which is extremely robust. It's robust to spatial heterogeneities, to temporal heterogeneities. And actually, one of the observations which I think is very intriguing, one of the things that's emerged from the study of hematopoiesis is that the multipotent progenitor cells, which are thought to be downstream of hematopoietic stem cells, those multipotent progenitors have a very large renewal potential. And one thing that's very striking is that if you ablate those cells and ask about their recovery, you might think that all the recovery would come from hematopoietic stem cells contributing to that compartment. But what you find is that most of the recovery, almost all of it, comes from these multipotent progenitors, these actively migrating multipotent progenitors in the bone marrow. And if you look at the nature of that recovery, it's also oscillatory. It has the same kind of oscillatory dynamic. And again, my guess is that the multipotent progenitors are regulating a near balanced renewal by precisely competing for diffusible niche factors in the environment of the bone marrow niche. So yes, I think it's pretty generic actually, but I'm sure I'll be proved wrong, but I think it's pretty generic.
The second question I had is mechanical feedback. Can you replace the chemical feedback by mechanical?
Yes. What a brilliant question. And the thing is, Jean-François, the thing is that this is a question I should be asking you. Now, let me say it this way. You may see that the feedback mechanism separates differentiation licensing from commitment, right? So there are two layers here. One is that you have to control the expression of differentiation licensing factors. And the second is that you then have to control the factors that promote commitment. It's a two-stage process. So I will allow you to have mechanics for one of those processes, but you can't have it for both. Okay. So this is what so I don't believe that mechanics alone can explain this mechanism. I think it plays a fundamental role in regulating tissue homeostasis in squamous epithelia as it does in all epithelia. But I don't think that mechanics alone can provide a mechanism to regulate stable density homeostasis. I think it has to be something else. And I think that's biochemical. Maybe autocrine factors, it may be paracrine factors, but I think it has to be biochemical.
Est-ce qu'il y a d'autres questions de gens qui regardent sur Zoom ou pas? Sorry, I'm asking them in French. No, they're quiet. Pas de questions ici? Okay, so thank you very much, Ben. I enjoyed it a lot.
Thank you. Thanks for listening. Thanks a lot.